So the next speaker will be Dr. Kelly Sullivan. He's an assistant professor in pediatrics developmental biology at University of Colorado. Dr. Sullivan graduated with a bachelor degree in biochemistry from Colorado State University and earned his PhD in biochemistry and biophysics from the University of North Carolina at Chapel Hill. And he did a postdoctoral training at the University of Colorado Boulder. Dr. Sullivan's lab focused on Down syndrome, and one of his current projects focuses on deciphering the role of interferon signaling in Down syndrome using a combination of cell-based and in vivo model. So thank you, and Dr. Sullivan, the floor is yours.
Is that on? Oh, hooray. Okay. Yeah, it's really a pleasure to be here. I'm gonna link that up a little. Thank you to the organizers for the invitation. I'm really looking forward to sharing some of our work on interferon signaling in Down syndrome. Clearly, this is going to be a bit of a departure from what we've heard today. I am not an expert in hearing or hearing loss, but I will touch a little bit on a new project, collaboration with some individuals in this room towards the end of my talk. Okay, so maybe. So I'm going to share just kind of some highlights from a journey that we've been on for the past decade or so. And I would be remiss if I didn't start by acknowledging a huge team that has contributed to our work. I want to mention Katie and Paula, 3 postdocs that led a lot of the animal work I'm going to talk about. Ross and Kendall, 2 super talented technicians, did a lot of our mouse phenotyping work. Mike and Lauren, members of my lab, talented molecular biologists. Our lead data analyst, Dr. Matthew Galbraith, and of course, my longtime collaborator and mentor, Dr. Joaquin Espinosa, who's the executive director of the Cernic Institute. Man, I'm cutting out, I'm sorry. Okay, so Down syndrome, probably everyone knows, is caused by trisomy 21, so that's 3 copies of chromosome 21. Down syndrome really presents as a kind of unique constellation of different clinical conditions. So we know that basically everyone with Down syndrome is going to have full-blown Alzheimer's pathology by the age of about 40. And that one. Try this. I don't know if that's any better. I don't think that's on either.
It should be.
Okay, well, I'm gonna go. So full-blown AD pathology by the age of about 40, different ranges of cognitive deficits, increased autism, epilepsy, pediatric hematological conditions, autoimmune disorders, OSA, pulmonary hypertension. This is just kind of the top of the list of co-occurring conditions that are far more frequent in the population with Down syndrome than the typical population. On the flip side, they're actually protected from some conditions, including a bunch of the kind of leading killers in the West. Coronary artery disease, high blood pressure, most solid tissue malignancies with the exception of testicular cancer. So it's really an interesting condition from that aspect. And another way to look at this is that basically every tissue system in the body is affected by Down syndrome, neurodevelopmental, cardiovascular, again, immune dysfunction and autoimmunity, which I'm going to come back to a bit later on. And then from the sensory perspective, obviously, we have conductive and sensory neural hearing loss, which again, I will touch on just a little bit, but not my area of expertise. What we don't have a good handle on is how we get from 3 copies of chromosome 21 to Down syndrome. You know, I would argue there's really one gene on chromosome 21 that's been pretty definitively linked in pathology, and that would be APP, the amyloid precursor protein gene on chromosome 21, and that seems to be sufficient to drive that neuropathology associated with AD, although I would be remiss if I didn't mention the fact that despite the fact that there's neuropathology by the age of about 40, the onset of dementia is highly variable. Only about 2/3 of people are going to, with Down syndrome will go on to develop dementia, and that has a window of onset that ranges by about 25 years. And so there's still a lot of inter-individual variation that we don't fully understand. So how can we study these kind of complex cause-effect relationships between trisomy 21 and pathophysiology? I'm going to talk about two of the tools that we use today. One is primary human samples. The other is animal models of DS. Again, this is a really complicated set of conditions with interacting tissues and a lot of different signaling. So we think that integration of data from a variety of sources and experimental models can really accelerate our understanding of these conditions and hopefully identify some putative mechanisms of pathophysiology so that we might be able to intervene and improve some of these conditions. Okay, so I'll start with just a little bit of an overview of some of our work in primary human samples. So we started something about 8 years ago called the Human Trisome Project at the Zurich Institute, which is a massive omics cohort with a lot of clinical metadata. So this all starts with individuals coming into the clinic, filling out a participant survey where we ask about many of these more commonly occurring conditions, and then getting an EMR and trying to extract what we call a digital phenotype. Then we take a blood draw, and from that we can view a whole bunch of different things that allows us to kind of map molecular phenotypes that we try to associate them with these co-occurring conditions, to see some biosignatures and understand what might be some of the underlying etiologies of these. So again, I'm a molecular biologist by training. I come from a cancer biology background. So we started with a very simple set of experiments where we just asked, what is the impact of trisomy 21 on the transcriptome? We got 6 pairs of age and sex masked fibroblasts, half of which have Down syndrome. And we just cracked them open. We took all the RNA and we did sequencing. At the time we did this in 2014, this was the largest RNA sequencing experiment that had ever been done for Down syndrome, which is kind of wild. But we made a couple of interesting observations right off the bat that set a couple of things straight in the field. One is that the genes on chromosome 21 are actually all upregulated. These had been done with some very underpowered studies in the past, and people weren't certain whether the genes were all up or down, or there was a difference. And you can see here from this Manhattan plot that all those red dots in chromosome 21 are up. The other main observation that jumped right out is that the vast majority of the differentially expressed genes are not on chromosome 21. So if we're only looking at the 400 or so protein coding genes on chromosome 21, we are missing the majority of the changes that are happening here. So what we're really seeking to find out here is, what is the signal amplifier, right? We start with maybe 12% of these genes from chromosome 21 that are differentially expressed, and this propagates throughout the genome to where we have now the other 90% of these genes that are differentially expressed. How do we figure out what that blue circle is, what's driving it? For this, we use a tool called Ingenuity Pathway Analysis, with a focus on a sub-tool called the upstream regulator analysis that really focuses on kind of transcriptional regulators, kinases, things like that. And what we found was a massive enrichment for interferon signaling. So basically everything here that's in orange, those things that are predicted to be activated based on the transcriptional changes that we saw. And what you can see here, basically everything in orange is interferon related, and it turns out all the things in blue are actually negative regulators of the interferon pathway. So the entire top of this list that we got are all interferon genes. And this signal is all coming from genes that are not on chromosome 21. So again, if we're focused only on chromosome 21, we're missing the entire story of what's changing in these individuals. And so what we get here, the things that are predicted to be activated in orange, again, are these interferon ligands, these IRFs and STATs, which are transcription factors. And the reason that we are getting these signals as predicted activators is because we have upregulation of all of these interferon-stimulated genes, or ISGs. Again, a few of these are on chromosome 21, so every once in a while we'll get a little bit of a faint signature, but the vast majority of this is coming from other parts of the genome. So that was in fibroblasts. We looked in lymphoblastoids, we looked in primary monocytes, we looked in primary T cells. And what we found over and over and over again was this elevated interferon signaling. We actually calculated interferon scores. This is actually a way that this is tracked in the fields of like lupus. Very difficult to measure type 1 interferons directly. So what people do is actually look at aggregate numbers of genes, and you can see these elevations. And just for comparison, this is the same score in individuals with COVID, and you can see that we see similar levels of elevation of these. Everyone in our cohort study here is not ill as far as we know. We ask extensive questions about whether they have been sick. We ask them to contact us if they get sick after the fact. these individuals are perfectly healthy, but they have this elevated interferon signal. So what is interferon? I don't think I have to explain this as much as I did before COVID. This is an antiviral defense system. It's a key aspect of immune signaling. It acts in every cell in your body, and I'll touch on this again in just a moment. Importantly, though, exacerbated interferon signaling is known to cause autoimmunity and a bunch of the other conditions that are co-occurring in the population with Down syndrome. So the way that we think about this is that people with Down syndrome are undergoing kind of a systemic sterile inflammation all the time. And as we were working this up, we started to realize there were similarities with another group of diseases, the so-called type 1 interferonopathies. So these are mutations that result in overproduction of the interferon ligands themselves. But what we see in common here is that these ligands are produced, they signal through the receptors that I'll touch on in just a moment, and the bulk of the readouts, the signal and the pathology, man, we are struggling with the technology today, are these ISGs, and that's exactly the same signature that we're seeing in the population with Down syndrome. So why is this signaling activated? Turns out that 4 of the 6 receptors that are encoded in the human genome are on chromosome 21, and that includes both of the type 1 receptors. These are expressed on every nucleated cell in your body, so you can imagine this has pretty wide-ranging consequences when this is overactivated. And then 1 of the type 2 and type 3 receptors, and again, the ultimate outcome of this overexpression, we think, is the transcription of these interferon-stimulated genes, and those we believe are contributing to many of the phenotypes that we see in the population. So just to show a little bit of the data that we found that was driving this, again, these 4 receptors in green here are the ones encoded on chromosome 21, and you can see these are overexpressed in these individuals with Down syndrome. Importantly, there's a lot of inter-individual variation, right? And we think this contributes again to these differential manifestations that we see in the population. So what one might expect if we have overexpression of these is that if we stimulate with those ligands, the trisomic cells would be more responsive to those, right? And that can be potentially quite detrimental. So it turns out that's the case. So if we just look here at dose response studies for euploid and trisomic cells with interferon alpha, you can see here the trisomic cells make a lot more of these response genes. MX1 is the viral restriction factor that happens to be encoded on chromosome 21. So you can see here it's actually elevated at baseline, but then it goes up even further. IDO1, ISG15, phosphorylation and activation of the STAT1 transcription factor. The whole cascade is hypersensitive to type 1 and type 2 interferon signal. When we looked at the cell surface expression, so these are immune markers, of the interferon receptor in about 100 different immune cell types using CyTOF. We found in basically every cell type we looked at, it's upregulated. And importantly, when we look again in these primary immune cells, we see that these cells are hypersensitive to stimulation. So here we did ex vivo stimulations of euploid, again, in trisomy 21 bulk cell populations via CyTOF, and we looked at the activation state of by phosphostat1 in CD8 T cells. And you can see here differential activation. When we look at NK cells, we can actually see that there's elevated activation of these NK cells at baseline, and this is further exacerbated when we stimulate with the interferons. And so that's what I'm going to show for our human data. We've been on this road for about a decade now. We've published a bunch of papers, but I hope that's enough to kind of set us to where I want to go, which is what are the functional consequences of having this hyperactive interferon response, and how can we test that? And so we have primarily used mouse models to get at this and ask the question, how does this interferon receptor triplication impact the in vivo immune response? Again, so now we're going to move into these mouse models. Very difficult to model a constitutive human trisomy in a mouse. They do not have the same genomic architecture that we do. In fact, the genes on chromosome 21 are spread across 3 genes in the mice, 16, 17, and 10. Thankfully for us, the largest orthologous region is actually on chromosome 16, about 120 of 300 or so protein coding genes. And that happens to include our 4 interferon receptors here in this so-called DP16 model. And that's the one I'm going to talk about. Notably, these mice exhibit a bunch of hallmarks of Down syndrome, congenital heart defects, developmental delays, cognitive deficits, craniofacial anomalies. So we think it's a really good system for us to kind of try to unpack what the differential contribution of these genes is. Notably, this interferon hyperactivation is conserved in these mouse models, and it's present across tissue. So just a couple of examples here via a heat map. You can see these are genes associated with the interferon gamma response that are elevated. Again, everything here very red compared to the blue, the wild types, these EP16s. And you can see that in all of the tissues, heart, lung, liver, brain, we see elevation of this signaling cascade. So again, one can imagine that might have pretty profound implications for the development and function of the animals. When we look again in their immune system, we see that the receptors are overexpressed, excuse me, we're here out the ratio of the DP16 to the wild type, and you can see everything here is to the right of this axis, which tells us that there's more cell surface expression of these receptors, and that leads us to test then whether they're hypersensitive, and in fact they are. So if we do again ex vivo stimulations with interferon alpha, now you can see here that the DP16 mice have more phosphorylation activation of STAT1. That'll lead to subsequent upregulation of those ISGs, which are not good for your body to have on all the time. So we asked what happens if we stimulate this interferon signaling systemically, and to do this, we treated with a little viral mimetic called poly-IC. This is just a double-strand RNA that the cell sends as a virus uncoding in your cells. This signals through TLR3 to produce type 1 interferons and other pro-inflammatory cytokines, and you can see that here. We make a lot of interferon that's not really detectable at baseline. You can see here in the wild type in black and the DP16 in green. We've massively upregulated these. So what happens to the animals? Well, it turns out the animals lose quite a bit of weight. So we have a pretty profound cachectic response. Again, you can see here the wild types lose a bit. The DP16s lose a lot more. In fact, the DP16s lose so much we have to take them out of the study. And some of these, in the early days, before we knew what we were looking for, we found them dead in the cage because they just lost so much weight. But over time here, you can see over about a week, we lose the vast majority of the DP16 animals just due to this kind of massive weight loss. So what happens if we normalize the copy number, right? We have 3 copies of all of these genes. We think they're driving a lot of this pathology. How can we test it? What we did, because all 4 of these genes exist in one cluster, and we weren't sure which of them were driving pathology in which tissues, we just deleted the entire 192 kilobase locus, and you can see here we got pretty clean deletions. This was a pretty ambitious knockout when we made it. One of the largest CRISPR-based knockouts, because we've been working on this for about 6 years at the time that we made it. And what that allows us to do is cross our knockout. So our locus here is indicated in orange. And you can see here the DP16, as we expect, that's got 3 copies, part of it's that segmental duplication. And then we have our knockout over here, where we have it's gone off of this allele. We cross these together, and what we get back our mouse model of Down syndrome, we get back our heterozygote, we get back our DP16 2X IFNRs, we call this lovingly norm, because that's normalized copy number. Then we get back our wild types, right? So within the litter mates, we have a fully controlled experiment here that we can look at, and we can ask, what is the impact of rescuing just the copy number of 4 genes, right? They're still overexpressing the other 116 or so protein coding genes in this. And so if we just look at a volcano plot here, again, we're looking at the fold change here on the x-axis and the negative log of the p-values on the y. If we look at the DP16 versus wild type, we have in the mesenteric lymph nodes, we have pretty widespread differential gene expression. You can see here a characteristic pattern of the triplicated genes in blue, where they're all up by only a little bit, but they're highly significant because they're all up by exactly the same amount, kind of 1.5-fold. We see the same pattern for our norm animals, but you can see the red dots here indicating the interferon receptor genes. Those are now gone because they're not significantly overexpressed. And when we look at the comparison of the norm to the DP16, the only thing that's significantly different is the interferon receptors. And that was kind of a bummer at first because we thought, wow, we didn't really fix anything. But when we looked more closely at the data, what we found was really subtle shifts on a global scale, right? So these didn't rise to the level of statistical significance for an individual gene. And here we're looking at the triplicated genes, and you can see not really any effect of the triplicated genes, which we wouldn't expect in either the lymph nodes or the brain. But when we look at all the rest of the differentially expressed genes and the absolute value of those changes, we can see here that these are severely diminished on kind of the global scale, right? And so what we're seeing is that many genes are changing by just a little bit, right? And so that allows us to kind of ask, what are the consequences of fixing all of this stuff by just a little bit? So we started ticking through some of these kind of hallmark phenotypes. So I showed you that if we put poly-IC on these animals, again, the DP16s here are in blue, they lose weight and they die. When we look at what happens in the interferon receptor normalized animals, you can see in pink here that they basically overlap with the wild types now. So we've completely rescued that poly-IC hypersensitivity. When we look at some of the more classical phenotypes, so here we're looking at congenital heart defects, right? Specifically, we're looking at ASD and VSD, right? So we have these openings now between the ventricles that shouldn't be there. In the wild types, we see a very small amount of these. In the DP16s, we see elevated levels. And then when we look again, these interferon receptor normalized animals, we can see that we rescue those, we bring those levels back down. When we look at developmental milestones, right? So these are very early neurological phenotypes, things like surface writing, ear twitch, eye opening, auditory startle. When we look at the DP16 versus the wild type, you can see that these are to the left of the dash here in the hazard ratios. That means they have delayed acquisition of the surface writing, ear twitch, and auditory startle. When we look at the effects of the normalized mice relative to wild type, those are no longer significant, right? And so we're pushing these animals developmentally back in the right direction. When we look at cognition, so here we're looking at the contextual fear conditioning. So this is where we put our animals into a little box here, and we give them a little foot shock, and we're measuring how much they respond to that, right? And so normally when we put the animals in, they move around a lot. At 3 minutes, we give them a shock, and you can see here the wild types, they start to freeze, right? They're realizing this is a bad place for them to be because something's shocking their feet. We shock them again, and you can see now that the wild type animals are freezing about 50% of the time. This is massively diminished in the DP16 animals. They're not learning that this is a bad place. They just continue to move around. We look at our normalized animals, and you can see that they're kind of an intermediate phenotype. So again, we're not rescuing everything here. We're partially rescuing it, which is, I think, what we would expect, right? This is a very complex polygenic phenotype that we're looking at in Down syndrome. And then they basically remember this the next day. And over time, the DP16s are starting to remember, and they're starting to realize that this is that bad place. But really no difference between the wild types and the norms and where they were on that first day. And finally, and I think probably most stunningly for me, we were actually able to ameliorate some of the craniofacial abnormalities here. So this is a partnership we had with Dr. Tim Cox, who did this micro-CT landmarking for us. And again, you can see, I think, pretty clearly here some differences between the wild type and the DP16, the more domed calvarium. It's got a shorter actual skull structure. And I mean, even I can see that those things are actually rescued when we fix the interferon receptor copy number. But when we do the landmarking, you can see here, there are a bunch of these lines in teal throughout. Those are the distances where they were actually exacerbated in one direction or the other in the DP16 animals, and we were able to rescue those, right? So they went back in the correct direction, and that yields these skulls that look more like the wild type. So what we take from this is that the normalization of the copy number can ameliorate some of the hallmarks of Down syndrome that are present in this mouse model. I'm not going to run through all of these again because I just touched on them. But this leads to the kind of main question here. Is there a way to normalize the interferon response in people with Down syndrome? Turns out, yes, there are. This is the Janus kinase inhibitors. So just to refresh us, we have the interferon ligands signaling through those receptors. Well, the next step here is activation of these Janus kinases, JAK1, JAK2, and TYK2, all of which have myriad drugs now that are FDA approved to target them. And so we set out to test whether or not these might improve things in Down syndrome. One of the things that we observed from our clinical assessments through the Human Trisomal Project is a massive upregulation in autoimmune skin conditions, including alopecia areata, hidradenitis suppurativa, atopic dermatitis, psoriasis, and vitiligo. So we wrote a bucket trial to look at individuals with Down syndrome that have one of these 5 conditions. It's a phase 2 single arm single arm open label, 16-week treatment with TOFA, and a 2-week follow-up, adults 12 to 60. Again, the big thing we're looking at here is safety. That was our first thing. People with Down syndrome are historically excluded from clinical trials. We know they experience drug metabolism differently, but we really don't know what that means, especially with some of these new drugs. So safety, we don't want to do any harm. And then looking at immune dysregulation, immune skin conditions, and then just kind of pie in the sky impact on cognition and quality of life. And I'm just going to show one individual here. So this young man came in, again, high school. He's got alopecia areata clearly. So his SALT score, this is the severity of alopecia totalis. And this is basically 86% of his skull doesn't have hair on it where it should. That's what the number 86 means. After 4 months of treatment, this is what he looks like. He told the clinical team that he would like to be known as Ed Sheeran. Which we happily obliged, and he's still on drug and he's doing amazing with this. So looking at these SALT scores across, basically everyone did better. This one individual here, our very first participant, actually had alopecia totalis. So it's not the patchy loss, they have no hair anywhere on their body. And this young woman actually started to regrow her eyebrows and her eyelashes. And we think that if she had just been on the medication longer, she probably would have seen some pretty profound benefits from that as well. From the molecular perspective, again, if we look at these interferon scores, this is the same data that I showed earlier, where these are elevated in the population with Down syndrome. You can see our group here at baseline starts and looks representative of this population with Down syndrome. By 2 weeks, we've already seen these scores go down, right? So we're getting the molecular readouts that we want. And I'll just note here in the light green, this individual had their second COVID vaccine a week before they came in, and we can see a spike in their interferon response, right? So we're just turning this down. We're turning it down to the level of everyone in this room, right? We're not immunosuppressing these individuals. We're just turning things down. It's still functional. It's still responsive. In this individual, 16 weeks, they had a fever. But we needed to get them in so that we didn't have a deviation from our protocol. So they came in, and again, we can see that their immune systems are responding at least at the level of production of these milligrams. So conclusions for this part, Down syndrome can be understood, at least in part, as an interferonopathy. It's also a ciliopathy and many other things, and I think we need to be kind of humble in our interpretations of these. But genetic normalization of the interferon receptor copy number does moderate many of the hallmarks of Down syndrome that are present in the animal models. And JAK inhibition can improve the autoimmune conditions and a number of other things I don't have time to talk about in individuals with Down syndrome. So I just want to finish very quickly. There was an observation during our trial that piqued our curiosity about hearing loss. So auditory testing is one of the safety labs for the JAK inhibitor trials, and we just kind of anecdotally noticed that several of our participants, they had improved auditory scores post-treatment, and so we started thinking maybe there's something here. We reached out to Peter, who put us in contact with Gabby and Heather and a bunch of other people at Boys Town. And we ended up sitting down and talking about putting together a project and thinking about what this could be moving forward. So it turns out there are incredibly high rates of hearing loss in Down syndrome, as I'm sure many of the people in this room are aware of. Up to 80% of individuals with Down syndrome have some form of hearing loss. Many of the cases are transient and secondary to OME, which I'll talk about in a moment. High rates of mixed and sensory neural. This is a slide I stole from Heather, so I don't know what all of these words mean exactly. Early onset presbycusis, and importantly, much of the information we have was obtained from individuals who were institutionalized, which was the case up until about the 1980s. So those are not reflective of what the population looks like now, and there's a lot of questions that we need to ask and answer about how individuals with Down syndrome are hearing. So I just wanted to note that we can model some aspects of this in the mice, which is super important. This is something that Peter and I are working on. And so here we're looking at the DP16 mice on the 67 black 6 background. Notably, these are young animals. These are 2 to 3 before the onset of the age-related hearing loss. But we can see here that they have the elevated ABR. And then also basically 100% occurrence of otitis media, right? So here we have nice healthy tissues, and I don't think I need to point any of this out. It's pretty clear what the OM is, right? We have all kinds of these immune infiltrates throughout the ear. And so we're very excited. We just received about 2 weeks ago an award from NIH that has three main aims. One is to do deep phenotyping with Gabby and Heather and Kristen, two of whom are here today. And for us to do a lot of the work that we do, which is to basically do biospecimen and multi-omic and try to see if we can get some biosignatures that are associated with these. And then Peter and I are also working on using these animal models to really get at some of the mechanistic studies here. So with that, I already made most of my acknowledgments, but again, we're a huge team. I want to acknowledge Include. So this is the relatively new trans NIH initiative that finally liberated us from NICHD, where we had been for a very long time. And finally, just everyone with Down syndrome, we're very engaged with our community. We appreciate that they share their perspectives, time, energy, and their samples with us. With that, I will take questions.
Thank you. So yeah, it's not working, I believe, for me either. So I'm gonna try to be thorough. Online, Dr. Kumar. Oh, wants to be unmuted. So Kumar? Yeah, you can speak.
Okay, can you hear me now?
Yeah.
Okay, cool. Very nice talk. I do have a very basic question. I'm not an immunologist, but my understanding is interferon helps with the immune system, fight infections and other diseases, if I'm not wrong. What's interesting is otitis media is particularly common in children with Down syndrome. So the Down syndrome patients should have a hyper interferon activity, if I understood your talk correctly. I'd like your comment on that. So maybe the infection somehow in the middle ear is not, the interferon is not working somehow?
Yeah, so it's a great question. Again, I'm not an OM expert. There was just a paper, actually it's not out yet, it will be out soon, from another group at Anchutz that showed kind of differential microbiome that's present in the ears of individuals with Down syndrome that have OM, and so it's possible that there are differences in that. I would note that depending on where we are, certainly in the upper respiratory tract, having a chronic interferon state actually lowers the barrier to bacterial infections. And so it's possible that that's something that's playing a role here. I think it's also possible that the massive amount of kind of structural differences between the ear and the skull of individuals with Down syndrome and euploid individuals plays a role, but I think it's very much an open question why there's more OM in people with Down syndrome.
Yes, I mean, the part that you said about, if I got your talk right, so the Down syndrome patients have fewer solid tumors, and that kind of makes sense with higher interferon. I guess that's supposed to fight some cancers as well, if I'm not wrong. So it looks like it is active, higher activity interferon is helping, but I'm just curious about this. Perhaps in the mouse model, maybe you can challenge them and test them?
That is what we're planning to do.
All right, thank you.
There's a question.
So this is wonderful. So I have a question for did you look at the cilia in these animals, or will you look at the cilia? I'm just thinking for about otitis and pulmonary disease and all kinds of other things. The cilia sometimes are part of the problem.
Yes. So we haven't looked in these animals. I will say, at least for primary cilia, we know that they are partially defective in people with Down syndrome. So there are a number of cilia-related genes on chromosome 21, notably pericentrin, and its overexpression basically causes everything to kind of clog up at the base of the cilia. It blocks sonic headshot signaling, and it creates all kinds of issues. Whether that's the same exact stuff that's going on for stereocilia or multicilia, I don't think we know the answer to that yet. I certainly don't. But I think it's likely that those would be affected as well. And I think those are questions that we're very interested in understanding. So we have colleagues in Anchutz who are studying cilia function, although I don't, again, not necessarily in the stereocilia.
No, I'm thinking more about the otitis media, actually. People who
Oh,
have
in the-
immotile cilia have sort of lifelong otitis.
So,
So that's what I was actually
yeah,
thinking.
I think it's a great question. I don't think we know, but generally in cell culture, we see that there are deficits in cilia function. In some of the mouse models, this is also the case, but again, the genetics there get more complicated about where the genes are and what the kind of genetic interactions are.
All right, so online there is journal, you're unmute.
Thank you. A very nice talk. I have a question about, do you see any changes in the hair cell number in this DP16 mouse model? Or loss of spiral ganglion neurons in these ones?
I just don't have any information on that yet. Hopefully in a couple of years, we can come back and share some more of this with, Peter's work.
Thank
All
you.
right.
So I have a quick question. So my question is like, does the level of interferon receptor affect the level of interferon in the system? Or you see the response only because there are more receptors?
Yeah, it's a good question. I don't know the answer to this yet. So, we've done a lot of cytokine measurements and it really depends on the study. Some people see elevated levels of the interferons themselves. It seemed like interferon gamma is probably up a little bit, maybe some of the interferon lambdas, but they're not up enough, we don't think, to drive the signal that we're seeing here. Now that's in the bloodstream. We do have some evidence that there's elevated production of the interferons potentially happening in cell culture. What the mechanism of that is, I have some thoughts. I don't know. So I think it's still an open question. I think, what's clear is that having more of the receptor hypersensitizes them when the ligand is present. Now, where that ligand is coming from, whether it's normal production from stress throughout development, right, or normal kind of just infection, it's possible that that is taking care of most of it. I don't think we know whether there are some intrinsic changes in the cells that are driving overproduction of the ligands in addition to the receptors.
Thank you.
Thank you.
Do you anticipate JAK inhibitors can balance these interferon levels?
Yeah, I think they can. So again, I think that's. Go back, check it. We can't. Yeah, we actually think that that's exactly what we're seeing, right? At least at the transcriptional level, we've measured a bunch of the cytokines, most of the inflammatory cytokines that we think are downstream of the interferon. Those are coming down to normal ranges when we put on the JAK inhibitor for a couple of months. So we think that most of this can be tempered, at least the interferon and some of the inflammatory stuff with the JAK inhibitors. The question is, which conditions would benefit from having that lower amount of interferon?
That's what I was thinking, because if interferon is going back again into loop, and then again it will be coming out, and then ligands will be available for the activation. So how are we going to control that? Because JAK is only going to inhibit only a certain pathway.
So again, the interferons don't control their own production, right? So it's other molecules that are eliciting the production of the interferons that then signal through those receptors and turn on those interferon stimulated genes. So I think putting the inhibitors in there, that breaks the circuit. And we know that, again, from the molecular level, that's pretty clear that's enough to turn down that signal.
So I actually have a follow-up question on this. How early you think you can start JAK inhibitor?
Yeah, it's a good question. So the drug that was in that trial is tofacitinib. This is a JAK1/3 inhibitor. It's picked because it has the most safety data on it. That's approved for use down to 2 years old.
Okay.
And so we're just getting ready to launch our third trial that is going to look, first one that we have is going to look in the pediatric population and try to get this.
Yeah, I actually don't have any idea if that will be detrimental during development or not. Like, because usually patients who have fetus who are affected can know very early on. So I was wondering if you think it could potentially be used actually during pregnancy?
That is what we want to know.
Okay.
I mean, that's kind of the vision, right? If everything is safe as we step down, right? We started with these skin conditions where we thought it was going to work to test the safety profile. We're now, our second trial is looking at neurological conditions. We're now moving to the pediatric population. trying to be very, very careful about how we go. Again, the genetic models here would argue if we can get in there early, there's
Yeah.
a lot of things that we might be able to improve, right? And so there's a big question about this, right? And certainly FDA advises that pregnant women stop taking injections. So I think we have a bunch of work to do before we even contemplate that, but those are things we're very interested in getting ready to test in the mice for starters. There's a paper that came out after the Nature Genetics paper from our colleague Kunwa Song, and he showed that if you treat pregnant female mice with the JAK inhibitors, you can rescue the cardiac deficits there as well. And so it seems like this is a viable path, but lots of things are cured in mice.
That would be beautiful, though. Yeah. So we're going to have a short break of 10 minutes before the award presentation, and I hope we're going to be able to fix the sound problems. Thank you.